Next to formation of bilaminar embryonic disc. Formation of bilaminar embryonic disc. See beta, bilaminar. Bi means two embryonic layered blastocyst. Two embryonic layered blastocyst. Formation of bilaminar embryonic disc. Before this topic, here already we are discussing about decidua. Okay, now decidua. Let us discuss once again about decidua. Okay, just let it be like this only. So decidua means here it is a spongy, highly vascularized layer around the embryo. Highly vascularized spongy layer around the embryo is called decidua. Decidua. Before this, decidua. Just a little bit discussion about the decidua. See beta here, this is uterine wall. Please observe carefully. Uterine wall. Okay, this is disc, embryo. Here this is amnion. We will discuss later about amnion. Okay, this is embryo. Here this structure is embryo. And this is yolk sac. This is yolk sac, this is amnion, and here this is embryo. I will draw with another color. This is blue colored part, it is called embryo. Embryo. Okay. Now, this is trophoblast. Trophoblast. Okay, syncytial trophoblast and cytotrophoblast. Okay. Okay. Don't be confused here. We are discussing about decidua. Later we will discuss about the formation of bilaminar embryonic disc. Okay. See here, this entire dotted portion, it is called decidua, a spongy, highly vascular layer. Okay, na? Spongy, highly vascularized means which is rich with blood vessels. This is decidua. So, based on its location, here decidua is divided into three regions. Three regions are three zones. Here this is uterus or uterine wall. Next, here this is embryo. Embryo. We will discuss later about this amnion and yolk sac. We will discuss later better. Okay, it is not correct time to discuss about these two parts. Okay, this is trophoblast. Trophoblast. Okay, next, the decidual part which is present between uterus means endometrium and embryo. Endometrium and embryo, this is called decidua basalis. Decidua basalis. This is basal part for this embryo. This is the basal part of this embryo. That is why this part of this portion of decidua is called decidua basalis. Next, say here this is the lumen of the uterus. Lumen of uterus. Okay. See, the decidual portion which is present between embryo and lumen of the uterus. Embryo and lumen of the uterus. This portion is called decidua capsularis. Decidua capsularis. Okay, which is present between embryo and lumen of the uterus. Lumen of the uterus. Remaining part of the decidua, remaining part means here, this portion, entire this portion of the decidua, it is called decidua parietalis. Decidua parietalis. Okay, so in this way here, decidua at early embryonic stage. At early stage, before formation of extra embryonic membranes. That is why here I am not explaining about amnion, yolk sac. So before complete formation of these two extra embryonic layers, here embryo is surrounded by highly vascularized spongy layer called as decidua. Based on the location of this decidua, it is divided into three portions or three regions or three zones. Okay, now decidual part which is present between endometrial wall and embryo. Here, this part it is called decidua basalis. Next, decidual portion which is present between embryo and lumen of the uterus. Lumen of the uterus. Okay, so here this is the lumen of the uterus, beta. It is leftover part of the uterus. Okay, so this portion of decidua is called decidua capsularis. Remaining part of the decidua which is present between here, see these regions. These regions it is called decidua parietalis. Decidua parietalis. Okay, so it is very important, Nana. Here, three questions will come from this diagram: basalis, capsularis, parietalis. Okay, choose the correct statement given below. Choose incorrect statement given below. Here, decidual portion which is present between here and here. Here, it will ask like this. Okay, please copy this diagram. Right? Make it fast. It is not given clearly in our Telugu Academy textbook. That is why, please draw this. Okay, see here this is called blastocyst. No, implanted embryo is called as blastocyst. Here this is cytotrophoblast. Please observe carefully. This is cytotrophoblast. Cytotrophoblast. Of course, outer layer, this is synocytial trophoblast. Okay, there is no issue with this layer, right? This is synocytial trophoblast, cytotrophoblast. Here, this is inner cell mass, embryonic stem cells. These are embryonic stem cells. They are arranged like a disc. That is why it is called blastodisc. Right? Now, after completion of implantation, here all these formative cells are embryonic stem cells are arranged in two layers. Okay, these cells are arranged in two layers. Here, this is upper layer. This is lower layer. Okay. These cells are arranged as two layers. This outer layer, it is called epiblast. Okay, lower layer it is called hypoblast. Okay, here embryonic stem cells or formative cells are arranged as two layers. Upper layer is called epiblast and the lower layer is called hypoblast. See beta here, epiblast, basically these are columnar cells. Columnar cells. Hypoblast, cuboidal cells. Yes.
Epiblast is made of columnar cells.
Yes, yes. Of course, basically all are cuboidal cells. But
Okay.
when they are arranged in two layers, gradually they changes. These cells, cuboidal cells will change or modified into columnar and cuboidal. Of course, cuboidal as it is, there is no change. Okay. So epiblast layer, basically it is columnar cellular cell layer, and it is primitive ectoderm. Primitive ectoderm. In future, further it develops into ectoderm and endoderm and mesoderm. All extra embryonic, all germinal layers are embryonic germinal layers. All three germinal embryonic layers, ectoderm, mesoderm, endoderm, derives from epiblast only. Please remember, it is very important point. All three embryonic germinal layers, ectoderm, mesoderm, endoderm. These three layers derive from epiblast only. Okay, so this is primitive ectoderm. This is now primitive ectoderm. Just we will consider this layer as primitive ectoderm. Okay. Next to hypoblast, it develops into further extra embryonic mesoderm. Extra embryonic mesoderm. Okay, not embryonic mesoderm. Please remember here. Extra embryonic mesoderm. It is primitive extra embryonic mesoderm. Here extra embryonic layers are there. Extra embryonic layers means amnion, allantois, chorion, yolk sac. Like here, 4 extra embryonic membranes are there. Each membrane again possesses 3 layers: ectoderm, endoderm, mesoderm. So in that way, here hypoblast forms primitive extra embryonic mesoderm. Primitive extra embryonic mesoderm. Hypoblast forms. Okay. Next, epiblast. It is primitive ectoderm. Ectoderm, endoderm, mesoderm. Here these 3 layers are primary germinal embryonic membranes. Ectoderm, endoderm, mesoderm. These 3 layers will develop from epiblast. Epiblast only. Right? Now, here this is the lumen of the embryo cavity. Okay. Next, here these cells, hypoblast is pushed down by these epiblastic cells. Okay. So now this hypoblast is pushed down like this. Then it forms the wall of yolk sac. Hypoblast pushed down to form yolk sac wall. Now this is called wall of yolk sac. Yolk sac, and this is yolk sac. This is yolk sac. Okay. So this is hypoblast only beta. Hypoblast pushed down to form wall of the yolk sac. Right. Next, here the position of this hypoblast. Okay, so hypoblast push you down like this. Now the place of hypoblast is occupied by some cells which are formed from epiblast. Here epiblast, the cells of epiblast continuously undergoes divisions. So these cells descends and form a layer, and this layer occupies the place of hypoblast. So this process, it is called delamination. Delamination. Okay now. So here these cells are migrating to other places. Here these cells are migrating other places. Here hypoblastic cells are migrated downwards, and cells, new cells which are formed from epiblast, they are migrating to this layer. So this kind of migration of cells, it is morphogenic migration. Morphogenic migration. Okay, na? Movement of these cells from epiblast to the place of hypoblast, and hypoblast here descends down and forms the wall of yolk sac. So such kind of migrations of these cells is called morphogenic migrations, and here formation of these layers, it is called delamination. Formation of these cells is called as delamination. Delamination means here pushing down this hypoblast, and which develops into wall of the yolk sac, and here separation of these two layers means cells which are formed from the epiblast, which descends down, which occupies the place of this hypoblast. So this is called delamination. Delamination. And the movement of these cells, morphogenic migration. Morphogenic migration. See, but I here this layer which occupied the place of hypoblast, here it is called endoderm. Endoderm. Embryonic endoderm. Endoderm. Okay. Next, say here this is the this view. This is epiblast. This is endoderm, and below this yolk sac is there. Okay, now if we see from upper view, okay, if you see from upper angle, the disc, embryonic disc appears like this. This is lateral view. This is lateral view. Here this is upper view. Okay. Here this is lateral view. See better. This is epiblast. This is hypoblast. Okay now. So this is upper view. Means that side this is epiblast. That side endoderm is there, and yolk sac is there. Okay. So let us see later stages. First of all, observe this diagram. Please observe this diagram, because we are going to another diagram. Okay. So please now observe this diagram. Delamination, morphogenic migration, endoderm, wall of yolk sac. Okay. Hypoblast, which developed into the wall of yolk sac. Okay. Wall of yolk sac develops into the mesoderm of the wall of yolk sac. This is epiblast, right? Just observe that. What is next step? Gastrulation. This stage is called bilaminar embryonic disc, and the next stage, it is gastrulation. Gastrulation means formation of gastrula. here now just forget about the yolk sac. Forget about the yolk sac. Just let us discuss about epiblast. This is epiblast, and this is endoderm. This is endoderm. Okay, endoderm, and this is epiblast. And here outer layer trophoblast is there. This is trophoblast. Okay, endoderm. The endoderm. Here this is epiblast, trophoblast. So this is bilaminar embryonic disc. Bilaminar. Only two layers are there. Epiblast, endoderm. Okay. Next, in the formation of gastrula, just let us see from this view. This view. This is lateral view. Better. This is lateral view. Now let us see from other view. Here it appears like this embryonic disc. This. This embryonic disc appears like this through upper view, right? Next, on the middle of the middle portion of this embryonic disc, a primitive streak appears like this. A longitudinal streak appears like this. This is called primitive streak. Primitive streak, just like a line. Okay, now this is called primitive streak. Primitive streak. Primitive streak. Okay, now a middle line appears. It is called primitive streak. Right. Next, along with primitive streak, a groove appears. Just imagine this is a straight line. Then this line develops into a primitive groove. A primitive groove. Groove means a furrow, like a primitive groove. It develops into primitive groove like this. Primitive groove. Primitive groove. Okay, primitive streak gradually develops into primitive groove. Okay, where this primitive groove is formed? On the longitudinal, mid-longitudinal portion of the blastodisc, bilaminar blastodisc, right? Next, and this anterior part of this primitive groove, a shallow part will develop like this. This shallow part, it is called primitive pit. Primitive pit. Primitive pit. And anterior portion, this anterior portion gradually develops and swollen like this. This swollen part, it is called primitive knob. Primitive knob. primitive knob. Okay, it is called primitive knob. Okay, next at this lateral portion of this primitive groove, here some folds appears like this. Some fold-like structures appears. These are called primitive folds. Primitive folds. These are called primitive folds. Okay. See, if we see through upper portion, of course, on the longitudinally on the middle portion of the blastodisc, here a streak, a longitudinal streak appears. Longitudinal streak appears. This streak is called primitive streak. Primitive streak, just like a line. Okay. Then this primitive streak gradually develops into a primitive. Just imagine here, this is primitive streak. This line between between my hand, two hands, this line primitive streak, and gradually this streak develops into groove like this. Groove, just like a canal. Okay, primitive groove, longitudinal furrow-like structure, that is called primitive groove. Next, at this anterior region of this primitive groove, a shallow path appears. This shallow path, it is called primitive pit. Primitive pit. Okay. Then anterior portion of this primitive pit gradually grows in size. Okay. So this solen anterior part of primitive groove, now it is called primitive knob. Primitive knob. Okay. Please copy this diagram. And here lateral folds. Then lateral folds appears along with the this primitive groove. These are called primitive folds. Primitive folds, right? So this process, it is called gastrulation. Of course, further developmental stages also there. Before that, please copy this diagram. Okay, make it fast. This is bilaminar embryonic disc. Okay, next it develops into trilaminar embryonic disc. Instead of bi here, trilaminar embryonic disc. See here, this layer is hypoblast. This is hypoblast, which further develops into yolk sac. Yolk sac. Okay. Next, here the place of this hypoblast, it is replaced by the cells which are descended from epiblast. So this process, it is called ingression. Ingression means here the cells, epiblast layer divides into numerous cells. These cells descends downwards and occupies the place of hypoblast, and it develops into endoderm. Okay, so this is first embryonic germinal layer. Which one? Endoderm. Endoderm is the first embryonic germinal layer. Next, here continuously cells will come from this epiblast, and these cells occupies the space between ectoderm, epiblast and endoderm. Then it develops into mesoderm. This is called mesoderm. down. Okay. Tomorrow we will discuss in detail, but just let me show these three layers. Okay, mesoderm. Next, now remaining this epiblast. After formation of this mesoderm, this epiblast, it is considered as ectoderm. Very simple. Okay. After formation of endoderm, here between this epiblast and endoderm, here this space is filled with mesodermal cells, which derived from epiblast. Okay, so this is called mesoderm. Okay, now remaining part of this layer, it is called as ectoderm. In this way here, three layers, three primary embryonic germ layers will form. Three primary embryonic germ layers: ectoderm, mesoderm, and endoderm. Ectoderm, mesoderm, and endoderm. In this way here, formation of trilaminar embryonic disc. Just make a change here. Trilaminar embryonic disc. Okay. So here this is now triploblastic animal. It is going to develop into triploblastic animal. Okay. Right. Next we will continue in the next class. Beta. Right. Wait.